Hi all, today I'm going to present on the topic case study pertaining to principal component analysis in plant taxonomy. So the contents that we are going to discuss in the upcoming slides are objective, introduction, methods and materials, result and discussion and conclusion of the case study, which is all about a technique PCA, that is principal component analysis. So the case study that I selected is the morphometrical studies on black pepper, Piper nigrum, L. Principal component analysis of black pepper cultivars. So this is a case study that has been conducted by P.N. Ravindran, R. Balakrishnan and K. Nirmal Babu, and they were part of the institution, that is Indian Institute of Spice Research, Merikunna, that is in Calicut, Kozhikode. And this case study is all about morphometrical studies of black pepper, that is Piper nigrum, and how the studies are conducted, that is based on the principal component analysis. So we'll be dealing with this case study in detail in the upcoming slides. So the major objective of this case study is the implication or the application of PCA, that is principal component analysis. Here in this case study, this PCA technique, that is principal component analysis, is used for determining the nature and extent of the divergence among the different pepper varieties. So in that they considered 44 cultivars and 7 wild accession varieties of black pepper, that is Piper nigrum, and that 44 plus 7 will be constituting 51. So that 51 varieties were considered as 51 operational taxonomic unit, that is OTU, and this is how this case study was conducted. So the major objective of the case study is this. So now we can start with the introduction part. So in the introduction part of this case study, it is like the information regarding the principal component analysis and what they are going to deal in this case study. All those stuffs are given. And in the introduction part, the PCA is explained as a statistical procedure that helps to convert the complex data into the simpler data. So here in this figure, you can see the principal component analysis represented. So in this graph, it is actually a 3D kind of representation, which is a complex one, and this is converted into 2D so that it will be like more simpler for us. So the principal component analysis, it deals with the conversion of the complex data into the simpler data so that it is more easier for the analysis. And here in case of this case study, the morphometrical studies in plant taxonomy were done, especially in case of the species, that is the plant pepper, which is Piper nigrum. In that plant, the morphometrical studies of that was done with the use of principal component analysis, and the quantitative analysis of morphological features and measurements to classify and differentiate this plant species was done. So here the morphological features includes leaf size, for flower morphology, etc. So now we can move on to the next section, that is the methods and materials of the that is mentioned in this case study. So in this case study was actually conducted based on the principal method, that is PCA, which is the principal component analysis. So this was carried out using 22 characters that was recorded from the 51 OTUs, operational taxonomic unit. So OTUs is nothing, it is the different varieties of pepper, that is I have mentioned in the last slide. And the analysis was actually carried out by using BMDP program package, that is specifically BMDP Forum PCA, and that was developed at the Department of Biomathematics, University of California in USA. And the computer analysis was done at the Computer Center of the Carnegie Mellon University in Pittsburgh, that is in USA itself. So this case study was actually conducted during the time of 1997. And during that time in India, the PCA and the implications of the PCA was not that much relevant, or it is not found to be that much used in India. So that this institute, that is the case study that is conducted in the institute ISER, which is Indian Institute of Spice Research, they depended upon the institutions at USA for doing this case study. So talking about the BMDP program package that I have mentioned in the last slide, that is BMDP Forum PCA. BMDP means it is Biomedical Data Package. It is actually a statistical software that was a collection of the programs which was widely used in the field of biostatistics. And this was actually, firstly, it was used for the analysis of biomedical and clinical data. And BMDP forum PCA, it is specifically referring to the PCA, that is Principal Component Analysis module within the BMDP software package. So this could be used to analyze morphometrical data also that were collected from different plant specimens. And also here in this case study, this method is used for the analysis of the morphometrics of the pepper. So next section is the result and discussion section. From the case study, that is from the methods that they have employed, which is the principal component analysis. From that, they have recorded 22 characters from 51 OTUs, that is operational taxonomic unit. And in here, this table, it is shown that here in this table, you can see that the values, like here the 22 characters are mentioned here, and the variance is explained, and the cumulative proportion of the variance in black pepper cultivars is also shown in this table. So from this table you can see it is till the 8 principal component, that is till the 8. Here you can see a drastic change in the variance, and all the others are like 0 point values, so that it can be neglected. So only this up to this, that is the 8 principal components were considered for the studies, for the study, and all the others, that is from 9 to 22, it was neglected as the values are very small. So now here you can see the 8 PC, that is the principal components, and the leaf size index, leaf length, leaf breadth, that is the characters which are occurring to be similar were placed in one principal component. And the next one is PC2, in that leaf thickness, lower epidermal thickness, upper epidermal thickness, like that. PC3, the leaf length, spike length index, spike length and peduncle length. Then in PC4, the guard cell length and the guard cell breadth, all this were like grouped under different principal components, and that are this 8 principal components. And for this 8 principal components also, this variables were calculated, and that is shown, like they sorted and rotated loading for this 8 principal component in the black pepper cultivars as shown in this table. These are the some of the graphs that are obtained by this PCA analysis. From the table that I have shown in the previous slide, they have drawn this graphs. That is, they have constructed this graphs. So in this graph is a PC plot, that is a principal component plot that has been done for the finding out the dispersion of the OTU, that is operational taxonomic unit, and also to find out the divergence and the similarities among those varieties, that the 51 OTUs, that is actually the 51 varieties of the pepper. So in this first graph you can see it is denoting the dispersion of OTUs between first and second principal components. So in each graph they will be considering considering two PC, that is principal components. In this graph, principal component 1 is taken in the x-axis and principal component 2 is taken along the y-axis. So here you can see the 29. This is 29th OTU. So in the case study it is saying that this OTU 29 is Panniyur 1, that is a pepper variety. So from this graph we can analyze that this Panniyur 1 is having wide difference in the, like it is not at all very much closer to x-axis or y-axis, and it is having more distance from both the x and y-axis. So it is considered that this Panniyur 1 is showing more divergence from all other OTUs, and like that if we are considering 4, it is very close to the x. So here you can say that this fourth, that is the whatever the OTU 4, it will be having a close relation to the PC1, that is principal component 1. And in the next graph, in this graph also you can see they took principal component 3 and 4, and here in the x-axis principal component 3 were taken and principal component 4 were taken in the y-axis. From this also the similar divergence and relationship can be found out from the given 51 OTUs. And in this graph also, this shows the uh, principal component, that is in the x-axis the principal component 1 is taken, and in the y-axis principal component 4 is taken. Thus they have created many graphs like this. I have only given 3 graphs in this. They have created numerous graphs in this case study, and with that they have related the different OTUs, that is the operational taxonomic units, and from that they have found out the relationship and also the divergence between these OTUs. So after that, finally they have created a clustering also. Like if the OTUs are found very near to the middle part, then we can say that it is more related. So by that the clustering also has been done, and the PC1, PC2, in all that the clustering was done, and with that we can say that the value D, that is D group, is found in every clusters, so that by analyzing this D group we can find out the relationship also. By analyzing, it is another method, that is by analyzing this D group also the relationship could be found. So these results they have studied that this case study is particularly helping to recognize the underlying similarities among the different cultivars of pepper. And here you can see in Panniyur 1, as I mentioned in a graph, in this Panniyur 1 is an example, and here it formed to be a distinctly differentiated on account of more than one species, that is principal components, as seen in the PC plots. So it is found to be having different divergence, and similar situations as existing in case of Karimunda and Kudiravalli, Vadakken, etc. So probably they all also might have originated as natural hybrids in the past. And so the principal component plots show that the characters involved in the divergence of various cultivars. So the characters that have been involved in cultivars like Panniyur 1, Karimunda, Kudiravalli, Vadakken, all were analyzed by using this case study. So moving on to the last section of this case study, that is the conclusion part. In the conclusion part, it is saying that this is for the first time that the such a study by using PCA that has been conducted in the black pepper, and in a similar way, another study is also conducted with the same like pepper species itself, which is occurring in the Western Ghats, and it was shown that this all is showing that the PCA is quite useful in establishing the nature of divergence among the various species. And here the absence of random mating and free gene flow in pepper is leading to the isolation of small population, and this population might have been undergoing this divergence through segregation, chance natural crossing, and accumulation of mutations. So that is this in the, after this case study they found out that that this all as the reason for the origin of variability in the black pepper. So this is how this they have concluded this case study. So with this, I am concluding my seminar presentation also. So PCA is the principal component analysis as a very important technique that is in the upcoming future also. It is a very important principle, like it is a very important analysis, statistical analysis that could be used very frequently for identifying many features both in zoology and botany. It is having wide applications in every area, that is it is multidisciplinary. So with this I am concluding my seminar presentation, and these are some of the references that I referred for this seminar presentation, and with that I am concluding this. Thank you for your valuable time.